Good morning, everyone, and welcome to the EPA's Computational Toxicology Communities of Practice. I'm Lauren Gessner, a Student Services Contractor with EPA's Chemical Safety for Sustainability National Research Program. Today we'll be hearing from Dr. Elin Ulrich, a research chemist here at EPA. She'll be presenting on EPA's Non-Targeted Analysis Collaborative Trial, or ENTACT. Before we get started, I would like to ask all of our participants to please mute their lines. If you're participating by phone, you can do that by hitting #6. If you have any issues with the video or audio, please let us know in the chat box, and hopefully at the end of the presentation we will have some time to answer questions. So with that, I will turn it over to Elin.
Great. Thank you so much for having me today. We are going to talk a little bit about non-targeted analysis and exposomics to start with, and then I'll get into the details of the study design for the collaborative trial and then also some results. I'd like to give a big shout out to all of the co-authors on this presentation. They are also co-authors on a manuscript that we've been working on, and there's a whole bunch of acknowledgments at the end, so I'd like to not forget to thank all of those folks from the get-go. So to start off with, we're going to talk just a little bit about exposure science. I'm sure most of you are somewhat familiar with this topic, but just to make sure everybody's on the same page, why do we do exposure science, and why is non-targeted analysis a good idea to do it, to study it? So reason number one is to understand the causes of disease. There was a paper that came out in 2010 that estimates 70 to 90% of disease risks are probably due to differences in environments. We always think of things like nature versus nurture, whether it's a genetic disease or an environmental one, and this is showing that it looks like, in large part, a lot of diseases are being caused by environmental issues. So it's important for us to understand what those environmental stressors are that might be causing diseases, and so that's one of the reasons why we're very interested in that for exposure. Additionally, we've got chemicals all over the place, and there are thousands of new ones coming out all the time. They're in commerce, they're new ones, we don't have a lot of information about them, and so trying to figure out which ones we need to worry about is pretty important, and so another reason to understand what we are being exposed to. Just because there are so many chemicals, let's try and prioritize which ones are actually causing some issues. So a little bit about the exposome or the exposome, depending on the day, I say it both ways. It was initially defined by Christopher Wilde in 2005, and people have quibbled over the definition over time, but in general, the exposome includes chemical and non-chemical stressors from internal and external sources across all life stages. And this figure over here kind of describes that a little bit. You can see different life stages starting during pregnancy, through childhood, adolescence, all the way up to adulthood. There are several different omics techniques that we can use to help understand the exposome. And one of the omics in general have been sort of cropping up for the past, I'd say, 10 to 15 years, even some before that. The exposomics is a relatively new one, and so we've added that to the list of things that we're using. Because the exposome is highly variable across time and location and individuals and activities and all those other things, it makes the measurements difficult. And we're also looking across a bunch of different matrices. We're looking across time. It just makes it kind of difficult. So we're looking at those internal things related to exposure, like metabolism and hormones and inflammation in your body, external sources of exposure, and the ones that we're particularly interested in are the chemicals, because I'm a chemist. It includes things like radiation and diet for specific external, but also general external stressors like climate, just general daily life stress and education. Those are all what I would kind of consider non-chemical stressors, and those are included in the exposome. We've got a couple different strategies to try and characterize the exposome, a bottom-up and a top-down, and what we typically do is this bottom-up strategy where we're trying to measure chemicals in all sources over time. And this is not an easy thing to do, and that's the way we've decided to tackle it, and we're currently finding out that the variability across time is quite drastic, and so it makes for a very interesting and difficult problem. So I'm going to now launch into a little bit of background on non-targeted analysis, because that is the tool that we use to understand the exposome. And non-targeted analysis is a chemical analysis technique. The way we approach it is using liquid chromatography with high-resolution mass spectrometry, and we'll talk about that a little bit more as we go along. But just to give you a little bit of the basics, targeted analysis, we have to kind of start from square one. If you remember back to Donald Rumsfeld's famous quote, these are the known knowns. This is a small list of chemicals that we study very well, that we have chemical standards for, that we can quantitate, we have good QA/QC for. Unfortunately, it only covers about less than 1% of the exposome. And so if we're only using targeted analysis, we're barely catching the tip of the iceberg as far as what we're exposed to goes. We really can't solve 21st century problems of any type blinded to most of the exposure data. It's just not going to work. So there's two more approaches. The first one is called suspect screening analysis, or SSA. You'll see that on my slides. And these are the known unknowns. It covers about 5 to 10% of the exposome. And in this case, we use very large lists of chemicals that we're trying to screen against. We don't use standards for the instrumentation, so the identification is not always confirmed, but we can get very good lines of evidence that a chemical is the one that we're looking at in our samples. So that's one approach. And then the final approach is what we would call true non-targeted analysis, or the unknown unknowns. And this covers the vast majority of the exposome, 90 to 95%. If you start doing some math, that makes sense. And in this case, we actually use the mass spectrometer and try and figure out what the chemicals are from first principles. We have a spectra. We know which atoms go into making up the particular masses that we're looking at. And we can figure that out backwards, sort of, or compare against standards that somebody has potentially run before. Actually, you can't do that in non-targeted analysis, true non-targeted analysis, because we don't know that it exists. So this is one of those situations where you really have to look at the data and try and figure it out. A lot of times when we talk about non-targeted analysis, we're talking about anything that is not targeted. So that covers both suspect screening and non-targeted analysis, or true NTA. So just to give a few examples of the kind of research that we do here at EPA using non-targeted approaches, you can see a lot of different matrices that we're interested in, and some of the examples include exposure surveillance. What chemicals are in the things that we eat, drink, breathe, the dust in our homes, those sorts of things. Just what's in there. Knowing what's in there, then we can use that to prioritize chemicals. Which ones are relevant in these mixtures that we're exposed to? Things like exposure forensics, where we're looking at chemical signatures of exposure sources. So if, for example, we're looking at a dust sample and there's a bunch of compounds that seem to rise and fall together, what is similar about those chemicals? Can that lead us back to a source that we might be able to mitigate so that people are not as exposed to those chemicals? There's another really cool technique called effect-directed analysis, and basically what you do is you run a bioactivity assay against a bunch of different fractions of a sample, and whenever the bioactivity assay lights up, then you do the chemical analysis to figure out what might be causing that. So that's a pretty interesting technique. We've barely scratched the surface on that here, but it's certainly an area that we'd like to get into. And then biomarker discovery, what chemicals are predictive of health impairment. We haven't done a whole lot in biological materials so far, but that's another direction that we would like to move so that we can start to put the pieces together to, again, figure out what is causing health impairment and from the exposure standpoint. So now we'll move on to ENTACT, now that you've gotten a little bit of an introduction to non-targeted analysis. Back in 2015, we held a workshop here at EPA to discuss what was going on with exposure science and non-targeted analysis. It was a field that we had been working in for a little while, but wanted to get some experts in-house and hear how their experiences were going. About 200 people attended, both in person and by webinar. And we had a number of different sessions over the 2 full days and had a number of speakers and poster presenters talking about their non-targeted analysis research. It was really interesting. It was really informative for us. And that has helped launch some of the efforts that are ongoing now. At the end of a day and a half, we finished the workshop with a half-day discussion on how we could use some of EPA's resources, particularly from ToxCast, to move non-targeted analysis forward in the whole field and really make an impact on what we're doing. That half-day discussion was the genesis of ENTACT, and I'll get into some more details about that now. So some of the goals and objectives that we have for, sorry, we're experiencing some feedback because somebody has not muted their line. I'll try to ignore it and keep going. So this is a list of some of the main objectives that we have for the ENTACT project. We really want to characterize the current method performance characteristics. It's really difficult when you're trying to understand all of the chemicals in a sample to know whether you're doing a good job or not, because you don't know what chemicals were in that sample to begin with, because if you did, why would you analyze it in the first place? So we want to try and understand the percent true positives, the percent false positives, true negatives, false negatives, etc., and really understand how the methods are performing. We don't expect that we're going to be doing 100% great job on everything, but if we understand where our shortcomings are, that pushes the field forward, and we can try and start filling those gaps. To that degree, we want to establish performance benchmarks because, again, like I mentioned, if you don't have a set of known chemicals that you knew were in the sample, it's really difficult to figure out how good of a job you're doing. So this study can help us do that. We also want to establish benchmark methods as well. We've got a number of different folks enrolled that are using all kinds of different methods, and so that will help us in that particular objective. Reporting standards for studies using these techniques. At this point, a lot of times mass spectra might get published, but they're all graphics. And it's very difficult to try and incorporate that into a cheminformatics approach when everything is graphics and not machine readable. So what kind of reporting standards can help push the field forward? And these are all things that we talked about at that 2015 workshop. Additionally, we all agreed that we needed more compounds and spectra available for our mass spectral libraries. And we want to make that available to the participants of ENTACT as well as the research community at large. So I've got 2 examples down here at the bottom. On the left-hand side is some stats for a website called MZ Cloud. It is publicly available and run by a company called Thermo. When I created this slide back in October of last year, you can see the statistics for the compounds and spectra that they had available at that time and what it is now. And so they're ever increasing the number of compounds and spectra that are available. But if you notice, there's only about 8,000 chemicals in that database right now. And DSS Tox has over 760,000 chemicals in it, so we're barely scratching the surface with this mass spectral library. Another reference that we often use is with NIST. They also have tandem mass spectral libraries, and you can see over the past, I don't know, almost 15, 20 years, the number of compounds in those libraries is increasing, but again, we're talking about 15,000 compounds, not 150,000, not 750,000. So this is certainly evidence that we need more of these spectra available to the public so that we can do a better job understanding what people are exposed to and understanding what chemicals are in the samples that we're analyzing. So back to our workshop, some of the critical needs that were identified are listed here, as well as the resources that we provided. So I'm going to try and tie these 2 things together. People asked for tightly defined ring trials to evaluate their NTA methods and performance. And a ring trial sometimes is also known as a round robin. I tend to think of those 2 things as being very similar, where a group of folks all analyze the same samples to try and come to a consensus. And so that's exactly what we've done. We'll talk a little bit more about samples in a minute, but we've provided SOPs for sample handling, analysis of data return, again, trying to define things reasonably tightly, what procedures we used for creating some of the samples, as well as a host of samples that came from the ToxCast program. So EPA has a library of almost 5,000 compounds that we were able to pick chemicals from and know that they were included into the samples. We've also provided well plates that have single chemicals and the maps to those for that mass spectral library building that we talked about last slide. So this kind of ties back into the availability of custom-made spike samples for the ring trials. As I just described, everything was created once and then shared out to the participants. So very custom-made, very well-controlled, and actually a large number of compounds. We'll get to that in just a moment. We also knew that the exchange of comprehensive suspect lists would help enable interoperability. And so we've shared both the MS-ready DSS tox list and the ToxCast list. The DSS tox list, when we sent it out, was about 720,000 chemicals, and the ToxCast list was about 5,000 chemicals. Certain workflows in doing non-targeted analysis can handle different sizes of lists. We tend to like the really big lists, but other approaches need a smaller list because of the computational power needed to deal with that. We also shared the dashboard with folks. Lots of folks that are participating in ENTACT are using the dashboard for a lot of their work. And we also shared mole files for all of the ToxCast chemicals so that folks could do fragmentation and things like that to help with their evidence, lines of evidence. So in addition, a lot of folks that are participating in ENTACT are sharing their suspect screening lists with us, and those are being added to the dashboard as lists. So this interoperability and exchange of lists has certainly taken off since this trial started. Finally, we wanted to have a retrospective analysis of the data. And so the way this trial works is folks get their samples, they analyze them in a blinded fashion, they return their data to us, and then we tell them what was in the mixtures and in the samples that we've provided. So they get to do a retrospective analysis of their data. And once we have all the data back in-house, we will do a retrospective analysis of all the data combined as well. So we have been trying to sort of meet these critical needs that we identified back in the workshop and hope that we've accomplished most of them. So on to the samples. So there's 3 different kinds of samples. So we've labeled them as different parts of ENTACT. Part 1 is the 10 ToxCast mixtures. And when we created these, we told folks that there was somewhere between 100 and 400 chemicals in each mixture or substances in each mixture, and the exact numbers are listed here. This is an actual picture of the mixtures that we got. They turned the most interesting colors I think I've ever seen in a sample in my life. It was a virtual rainbow, and I thought it was really cool. So we've decided to use its photo here. Part 2 involves 3 standardized exposure-relevant extracts. And we decided to provide extracts so that there was very tight control over what people were analyzing. If you hand somebody a dust sample, they can extract it several different ways, and so what went onto the instrument for analysis might not necessarily be the same from lab to lab. So we decided to provide extracts. And the 3 exposure-relevant media that we chose were human serum that came from NIST. That's a standard reference material. We also chose a house dust, which is also a NIST SRM. And then we also contracted with Oregon State University to use silicone wristbands. And because of human subjects research, we asked them to do an outdoor air exposure with the wristbands. And in every case of these extracts, we have left one version of it unspiked and altered, and a second version of it fortified. So for each matrix, you get 2 samples. You can see from the mixtures which kind of mixtures go where, so you've got different numbers of chemicals in each of those matrices. Part 3 includes individual ToxCast standards. We have 2 different versions of this. One is for ENTACT chemicals, and there's about 1,300 of those with the individual chemicals on well plates. It turns out to be about 6 well plates. And then we've also provided ToxCast, the entire ToxCast library, to some of our vendors so that they can work up those mass spectral libraries for a fairly large number of compounds that we care about. So the whole reason that this is going on is because these are chemicals that we care about. They're important to exposure, and so almost 5,000 chemicals that could be added to a reference library is great progress. So a little bit more into these mixtures. The way that these came together was really the brainchild of Chris Grelke. We like to call him the evil genius behind these mixtures, and there were some aspects of this that are truly diabolical. I think he's kind of proud of that, and we are too. So you can see that the first four mixtures have 95 chemicals. Mixtures 5 and 6 have 185 chemicals. Mixtures 7 and 8 have 365 chemicals, and I will get back to 9 and 10 here in a minute. You'll notice that there's a tiny little blue bar down at the bottom of each one of these mixtures, and there are 5 chemicals that were added across every single mixture so that we could compare across the different mixtures. You'll also notice that in mixtures 1, 5, and 7, there's an orange bar, and that orange bar represents a set of 90 chemicals that were introduced for every size mixture that we have. So this helps us figure out whether increasing the complexity of the mixture impacts whether we can see certain chemicals in that mixture. So we wanted to include that. And it's also important to note that those were the mixtures that we spiked into the prepared mixtures into the prepared extracts that we sent out. That way we've got a class of chemicals or a list of chemicals that we can compare across all the different matrices. The chemicals that were in mixtures 1 through 8 are all grade A chemicals, and the ToxCast chemicals have undergone QA/QC testing prior to this, and in order to get a grade A, you had to have some evidence that the chemical of interest was in the sample, and for us that would include the correct accurate mass and a purity above 90%. So Chris made these mixtures so that they contained grade A chemicals, and so that there would be minimal overlap between isomers and isobars. Isomers have the exact same chemical formula. They're going to have the exact same mass in the mass spectrometers. That makes them very difficult to tell apart. Isobars happen when you have a different chemical formula, but the masses are doggone close. And those, if your instrument is not working at its optimum performance, might also be difficult to tell apart. So Chris was nice for about 8 of these mixtures and tried to keep things pretty calm, good quality compounds, not a bunch of overlaps. For the last 2 mixtures, he made them hard. And so mixture number 9 has a lot of isomers and isobars in it. And just kind of testing the methods to see how well we can do when we have that sort of situation. Mixture 10 is a much larger mixture, and here he's included chemicals that were not grade A. So it's more likely that the chemicals had some issue when they were purchased, that they were not of the highest purity, they might degrade, something along those lines. So he wanted to throw us a couple curveballs down there at the end with mixtures 9 and 10. So of those 10 mixtures off to the right-hand side, you can see there were a total of almost 2,000 spiked substances, but there were lots of replicates. And you can see those replicates, how they panned out over here on the side. There were a total of almost 1,300 unique substances. And for a ring trial like this, this is a phenomenal number of compounds. And without EPA's resources, I don't think this would have been possible. And Richard is the one who manages a lot of this kind of stuff along with Kathy Kutros, and they've spent years trying to acquire these chemicals and build up this library for testing with in vitro assays. So just a quick list of the ENTACT participants. There are a total of 28 at this particular moment. You can see the breakout by sector and by location, and I will mention that the folks down here at the bottom are all instrument vendors, and they have gotten the single well plates for the entire ToxCast list. I get confused between ToxCast and DSS Tox, so I still have to pause on that. So they've gotten that almost 5,000 single chemical list. In addition, Scripps got that same set of well plates, as well as the University of Alberta. Most everyone else received both the mixtures as well as the exposure samples. A few only decided to look at the mixtures. So now I'm going to dive a little bit into the results. This is a set of slides on the instrument methods. And if you're not a chemist, this might not be the most interesting, but I'll try to move through it fairly quickly. What I want to show here is the diversity of methods that are being used, but also some of the overlaps. It's important for us to have that diversity so that we can compare across many methods, but it's going to be really handy to be able to compare similar methods to see if they get the same results. So in this slide, we've got 2 laboratories that have done gas chromatography work and one group that has done a technique that is just not like any other. They're using an FTICR instrument that has a very, very, very high resolution for the mass spectrometer. And that's provided them with some interesting opportunities. In one case for the GC, it's a high-resolution mass spectrometer. In the other one, it's a low-resolution mass spectrometer. We haven't dug into the details to see what is working best right now, but it's nice to know that these differences do exist. This next slide covers the instrument methods that are liquid chromatography and time-of-flight instrumentation. You'll see an awful lot of Agilent QTOFs. Some of them are the same model, some of them are different models. Some of them are using the same ionization source, like electrospray ionization. 1 or 2 of them have thrown in stuff like APPI, atmospheric photochemical ionization, something like that. You can see we're frequently using similar kinds of columns. You'll see that the C18 columns are cropping up quite frequently, but every once in a while a different kind of chromatography column pops up. A lot of folks are using methanol or acetonitrile for their mobile phase. Ammonium formate and formic acid seem to be the most common additives. We have more liquid chromatography methods, but this time with an Orbitrap mass spectrometer. So we've got a whole list of folks that are doing that. Some interesting ones to note, on 10A, they are actually using ion mobility mass spectrometry. There's one other laboratory that's using that as well. This is an orthogonal technique. It's separation based on the shape of the molecule. So if you think of a piece of paper that you've crumpled up and a piece of paper that's laid out flat, if you drop both of them, one of them is going to get hit first, and it's the compact one. It's the same idea for the mass spectrometer, where the drift tube basically separates things based on their conformation and their size. So again, you'll see some of the similar things that I mentioned on the last slide, but again, we're using a totally different kind of instrument. Here we've also added APCI, atmospheric pressure chemical ionization. Sometimes the acronyms will get you. So again, just a good list of methods. At this particular point, we've had approximately 15 or 16 laboratories submit information to us for their blinded analyses, and maybe about half of those folks have submitted their unblinded analyses as well. So we're still having data trickle in. So let me walk you through this slide because it has a whole heck of a lot of numbers on it, but quite frankly, the colors are probably more important. So up at the top, we've got the different mixtures labeled 1 through 10. Right underneath that, we've got the number of chemicals that we intentionally added to those mixtures. And I say that very specifically because we don't know whether that's the actual number of chemicals that were in there. There are possibilities that laboratory contamination, impurities in the sample to start with, reaction products, degradation products could also be in those mixtures. We didn't add them, but they might be there. So you can see the number that we actually spiked in, and then compare that to the number that's in the table from the different laboratories. So Lab 1, Mixture 1, reported that there were 128 chemicals in that mixture. The colors then correspond to whether they over-reported in the case of the pink one, or like I just mentioned, that 128 in a mixture that supposedly had 95 things added to it. The green ones are about right, so their percentage of how much they reported compared to what we actually added was close. And then there were some folks that underreported, and those are shown in blue. One thing I want to caution people against is that these are all blinded results. These are how many things people told us were in the samples when they didn't know what was in the samples. So it's not really fair to start comparing methods just based on the number of things that they reported were in the samples. It gives us a good sense of what the data is looking like, but we don't know whether the number of things that they reported were right or wrong at this stage. So I want to caution people from picking out their favorite and saying, well, that method must be the best because they got all green. That's not the case. Different people reported their results differently as well, and there's large possibilities that the way I counted things might have double counted. They might have reported things that they just weren't quite as sure about, but they definitely saw. So there's a lot of variability in the way this data came back to us. So again, want to just caution people that these are blinded results. It has nothing to do whether they were right or wrong. This is just how many things they saw on the samples.
Can you go back one slide so we can see what instrument generated?
No.
14? Seriously, what's 14? What was the instrument?
That is an Orbitrap. I can tell you who it is, but I'm not going to. Yes. So it was an Orbitrap instrument, and I think the reason why those results are so high is just simply the approach that they took. And again, it may be a situation where I've double counted things and shouldn't have. So don't read too much into this. But one of the things I would like to point out that most of this table is pink. And that means that people are finding more things in these samples than we actually added. And they might actually be there, and that's really important for us to know because these chemicals that went into the mixtures were tested in vitro assays, the high throughput, well plate assays. If there's something else in that sample from the get-go, we need to know about that. And I think that these ENTACT results will start to help us understand what other chemicals might be in these mixtures and therefore back into the original chemicals that they came from that we've tested.
But the purity of those original chemicals was supposedly tested.
It was supposedly tested, and it was supposedly over 90%.
So there's 10% that could be...
There could be up to 10% impurities, but it could also be a degradation product. These chemicals have been in storage for quite some time. When they got tested on the in vitro assays, they may not have been degraded. They may be degraded now. We don't know. But it starts to help build the picture. And the more things freeze and thaw, the more types of things can happen.
I'm not a chemist, but when they tested it to get, say, it's 90% pure, did they use the same kind of instruments as these people are using?
No.
Not necessarily. They used LC and GC mass spectrometers, but not necessarily high-resolution ones. They may have had better or worse sensitivity. They weren't looking for everything in the sample. They were looking for one thing. And so they tended to use very quick chromatography methods just to get the compounds off so that they could tell whether the compound they were looking for was there or not. So Anne can tell you a lot more about that sort of thing, but there are lots of caveats with this data. We've really not dug into this hardly at all. This is something that's really easy to do. You look at a spreadsheet, you count how many rows are there. That's the kind of data you're looking at. So, like I said, I just wanted to caveat that with this is not any indication of the best method at this point. So we also wanted to describe the exposure samples. Same type of situation as the last slide where we've got dust, the unfortified and fortified, the serum, and the wristbands. We only know how many chemicals we added to the fortification. We have no idea how many chemicals were in the dust to begin with, so I put question marks there. So I've not color-coded the ones that were left unaltered. And when you look at the ones for the fortified, that also does not account for anything that was there to begin with. So these results are a little bit harder to interpret, but it does, again, start to give you some sense of what's going on within this data set. The biggest take-home message I got from this was most people have not reported on the exposure samples yet, and that actually includes our laboratory here in RTP. We're still working on those samples, but it's a little bit harder because there's a matrix involved. And so there's a lot more noise that you have to deal with, and it's just harder to tease apart. Again, you'll see a range of different colors in these cells, but in some cases people chose not to analyze them. In other cases they just have not managed to get the results in yet. And I think eventually we'll get some more results, but I expect fewer results than we did for the mixtures. Keep in mind, mixtures is sort of a best case scenario. We don't actually ever look at real samples from the environment that people are exposed to that don't have a matrix with them, that are dissolved in DMSO. That never happens. So it's a really good test of the instrumental methods and the kinds of compounds that we can see in a perfect world. This is real life, and it's hard. The next couple of slides, we wanted to dig in just a little bit, and I mean just barely scratching the surface, as far as what kind of chemical space we were covering. So this is a Venn diagram that describes some results where the laboratories had gone through and done a good job of their unblinded results. So they submitted their blinded results. We told them what was in there. They said, we got these right. So one of the two laboratories that is depicted here used gas chromatography. The other one used liquid chromatography in electrospray positive and negative mode. So we've combined those results, and out of the 1,269 chemicals, unique chemicals that were in the ENTACT mixtures, 195 of them were not detected by either method. And I did some quick math this morning. That's about 15%. And quite frankly, I think that's not too bad. I'm kind of pleased by that. It could have been worse. So that's a good sign. And it's possible that other folks that do GC or other folks that do LC will actually detect those chemicals. This is just two laboratories. Very preliminary results. The other thing that struck me as very interesting is that there are 378 chemicals that you can see by GC that you cannot see by LC. You can only see those chemicals, almost 400 of them, with the gas chromatography. For the LC, there's only about 200 of them. So to me, if I was starting off in this field, that would be very telling as far as which approach I might take on first, because I'm going to get more coverage of the chemical space that ENTACT covers, which certainly is not all of chemical space. But it starts to kind of show how things fall out. The other thing that I thought was pretty cool is there are 37 chemicals. It doesn't matter what technique you use, you can see them. It's not a very big number, but it's nice to see that there are a few of them. So that's a set of chemicals that as we get results back from pretty much anyone, we should expect to see. More on chemical space. This is kind of a busy figure, so let me walk you through it. Each panel is a different physical chemical property. So we've got things like boiling point, melting point, log KOW, also known as log P, water solubility, etc. Each scale is different, so it's in its natural space. These are box and whisker plots, and so the middle bar that you can hardly see in the room, but hopefully you can see on the webinar, is the median. The edges of the boxes are 25 and 75th percentiles. The edges of the whiskers are 10 and 90, and the stars at the edges are 5 and 95. The different colors correspond to different set of chemicals. The red bars are ToxCast, and everything else is a subset of ToxCast chemicals. So the ToxCast list is almost 5,000 chemicals. The next bar is the ENTACT chemicals. That's the 1,269 that we've been talking about. The next green bar is GC-only compounds. So you could only detect them by GC. That's the 378 that I mentioned in the last slide. The teal bar is LC-only compounds, and that's about 200 chemicals. And then the gray bar is those that were not detected. So that was the 195 from the last slide. One of the very important things to point out is that the values for these physicochemical properties are from the dashboard and the opera predictions. So these were not measured, these were predicted. And the reason we've done that is because the measurements are of different quality, and you cannot have measurements for a lot of the chemicals. So of the 5,000 in ToxCast, there were less than 100, maybe even less than 50 that we did not have predicted values for. So on to what this tells me. The first thing that I'd like to point out is I want to know what is it about the chemicals that we cannot detect by either gas or liquid chromatography that makes them not detectable. And so I thought, well, if we start looking at the chemical space, maybe that will give us some idea. And unfortunately, it doesn't. The gray bar overlaps with the green and the teal bars. So it's in the same chemical space for each of these properties that the GC and the LC compounds are. I will say that there are statistical differences between all of these, the distributions, but the chemical space that we're covering is at least somewhat similar. It's the distributions that are different. So that didn't really help. So what that tells me is there's something else about these compounds other than probably the chromatography, because we've covered as much chromatography space as we possibly can. So maybe it's something about the way it ionizes in the mass spectrometer that is allowing us not to be able to see it, or that's preventing us from seeing it. There's also a couple other results in here that don't necessarily make the most sense, so I wanted to talk about those first. So boiling point, we expect GCs to look at high boiling compounds. They're supposed to be volatile. Well, the median for the LC compounds is actually higher than it is for the GC compounds. So that doesn't make a whole lot of sense. So if we come down to the bottom and look at the vapor pressure chart, now we're making some sense. Even though the boiling point might be higher for the LC compounds, the vapor pressure is lower. Okay, that makes more sense. Another one that doesn't necessarily make sense at first glance is the water solubility. The LC-only compounds have lower water solubility than the GC-only compounds. And the reason we think that that happens is because we're using reverse phase liquid chromatography, where all of those very polar, very water-soluble compounds come out at the very beginning of the chromatogram in a giant batch that we can't tease apart. And so they're very difficult to identify when they all come out at the very, very beginning. So that's why we think that is happening. So again, you definitely see differences between the physicochemical properties and how the landscape plays out. Some of it makes sense, some of it doesn't, but we've got some more digging to do. Again, we're just looking at a couple of results and very initial results from what we've gotten back from ENTACT. In the next few minutes, I want to talk about some of the results from our own work. So EPA is part of this trial as well. So how did we do with the synthetic mixtures? Just a little flavor for this. John Sobis has a great hour-long presentation just on these results, and there's a paper that's been submitted on this. But I'm just going to give you a little bit of flavor. So here's the workflow that we used to actually analyze the samples, do the data processing, et cetera. And I'll walk you through that step by step. So we've got 10 synthetic mixtures. Each mixture was diluted at 3 different concentrations, and each dilution was run at 3 replicate injections. We also used blanks so that we can try and subtract out anything that was in the blank and discount that as a feature that was in a sample. The analysis was done on an LC quadrupole time-of-flight mass spectrometer that's upstairs in my laboratory using electrospray ionization in positive and negative mode. In this particular step, we're looking at MS1 data only. So we're not fragmenting the molecule and trying to look at very detailed mass spectra that would give us a good fingerprint and identification. We're getting an accurate mass for the intact molecule, which can help us back out to the chemical of interest. Later, we'll talk about the MS-MS results too. After we've gotten the data collected, we do peak picking and alignment in a piece of software from Agilent called ProFinder. After that, we do formula assignment, and we screen. In this particular type of analysis, we're doing suspect screening. If we go back to that slide with the iceberg on it, we're using a very large list of suspect chemicals to screen against. And so we take the data from the mass spectrometer, we see if the masses show up on the DSS tox list, and start to put those things together. The next step, we're actually removing some of the features. So we subtract the things that were in the blanks. Because we've done 3 different dilutions, if the abundance from the instrument doesn't change by that same fold, we can take those out as well. We do give it some wiggle room, so it doesn't have to be perfect. We also do a formula match score cutoff. There's some information in the mass spectrum that can tell us whether we've got the formula correct or not, and how correct it is. So we've got something that's 50% match that's not nearly as good as 90%. We're going to throw away the 50% ones. When we do this step, later on down the road, when we've been unblinded, we can actually start to look at our performance. And in this step, we can look at the performance based on whether we got the mass of the chemicals right that were in the mixtures and whether we got the formulas correct. At this point, we haven't figured out what chemical it is, so we're just looking at those 2 pieces of information. The next step in the workflow is to actually pick out which chemical we think it is. We use the CompTox chemistry dashboard for that. We do batch searching. I'll go over a couple of slides like that in a moment. And we use data source ranking. We aren't using a whole lot of information from the instrument at this stage. We're working on lots of different things like retention time and predicted mass spectra to enhance that characterization and which chemical we think we've actually found. And at this stage, when we're unblinded, we can figure out whether the chemical that was spiked in showed up in the top 10 or the top 1 substance that gets returned from the dashboard to tell us whether we got it right or not. And then finally, we can do some chemical structure corroboration. So we've got a list of chemicals that we're interested in because of the MS1 results that we've gotten. We don't know what they are necessarily per se, but we know what their masses are. So we can tell the mass spectrometer what those masses are that we're interested in, and anytime it sees those masses, to fragment that. And that gives us a whole nother set of information that we can use to try and pick out the chemical of interest, the one that was in the sample. We do that using Agilent PCDLs, which is a personal compound database library. It's just this library of mass spectra that we talked about early on in this talk. And so when we've been unblinded, we can look at those. And as we move down this workflow, we become more and more confident in the assignment of the chemicals that we think we've seen. I wanted to take just a quick moment to talk about the differences between DTX SIDs and CIDs. Many of you may be familiar with this already. I apologize if I'm boring you. I'll try and go through it quickly. What we see in the mass spectrometer is not necessarily the exact chemical that was spiked in. And so there's a conversion to make it the structure that we started with MS ready. So we convert it from what it starts off with, its actual true chemical structure, into what we would see in the mass spectrometer. And so this is MS ready processing, and it goes from a DTX SID to a CID. So for a couple of compounds that did get spiked into the mixtures, these are stereoisomers. When we do the MS ready processing, we take away stereochemistry, we take away salts, and we take away components or solvents of hydration. So that makes the structure MS ready. It's very similar to the processing that gets done to make a structure QSAR ready. So these two are stereoisomers of each other. If you look at the formula for those compounds, what we detected on the mass spectrometer is this formula right here. If you do a dashboard search, you get 4 different DTX SIDs. If you then convert that to the MS ready form, 2 of those structures wind up with the same DTX CID that would correspond back. So what we actually see in the mass spectrometer correlates to the CID. We can then kind of point that back to the SID and say it's one of these 2 because that's how it got to it. We can't tell the difference in the mass spectrometer. So I wanted to kind of go over that just a little bit because when we do our did we get it right or not, we're looking at the CID level because that's what the mass spectrometer produces for us. I'm going to try and switch over to the dashboard. That looks like it. So I wanted to just go over a couple of things that we use in particular from the dashboard side because this is stuff that's very mass spectrometry centric, and so not everybody may have used it. So on the advanced search tab, you can do mass searches, so you can type in an accurate mass, plus or minus a particular area, and see what comes back. You can also put in a formula search. So if we've done the DSS tox matching with our data, it returns a formula to us. We can just put the formula in. We can also generate formula. If we've got a mass that came off the mass spectrometer, we just don't know what it is, we want the dashboard to tell us, we can look that up as well. We also use the batch search quite heavily, and I'm going to show, we're going to type in a monoisotopic mass, and I'm just going to make one up, 543.976, whatever. So once we've done that, we can start to download the chemical data. The important part of this is the kinds of data that we can get back. So we can get all kinds of chemical structure information. We can get MS-ready information to help us match up. We can get opera model predictions. So this is how I downloaded the lists that you saw in a previous slide. We can get data sources. That's how we do our ranking. We can look and see what kind of lists these chemicals show up in. So maybe it's a drinking water list. Well, by golly, if I'm looking at drinking water and it's on a drinking water list, that's another piece of evidence that that could be the chemical I'm looking for because somebody else has already found this chemical in drinking water. So that's the kind of data that we tend to retrieve. Let me see if I can figure out where I was. This is where I was. So that's what's on this slide is the actual dashboard information. I just felt like it was a little bit hard to see on the slide, so thought I'd actually do sort of a live demo for that. Once you've input data, for example, in the batch search, you'll get a spreadsheet back. You can download it a couple different ways. The nice thing here is you can see I've kind of grouped these things together with the outlines. You can see all the different chemicals that might potentially match. And you can get information about the data sources, again, to help rank, but you can also get information from ToxCast testing as well. How many assays was it a hit in? Well, that's a chemical that I'm a little bit more concerned about now because it might be toxic. So we use the dashboard in this way, and it's been very, very helpful, and we're very thankful to the entire dashboard team for listening to us and helping implement all the things that we really wanted it to do. All right, back to our results. Again, this is a kind of busy slide, so let me walk you through it. We've got the mixtures going down the side. I've got the number of spiked substances listed in the next column. And we talked about isomers and isobars before. These are somewhat difficult to tell apart in the mass spectrometer, particularly the isomers. The accurate mass for those is identical. So when we talk about adjusting things in a moment, we're discounting isomers because we have a really hard time telling them apart with the mass spectrometer. The isobars we can tell apart if everything's working well. So the next set of columns are our blinded results. So we've got the number of chemicals that were true positives, the number we got right. And this goes back to some of the information that we were looking at on the workflow as to, at the point of being blinded, whether it was right or not. So if it ranked very highly in our dashboard search or we had confirmation from MSMS information, we considered that a true positive. Next, we've got a column for true positive rates, and then the column after that is an adjustment of that for the isomers. So we've basically just taken the isomers out. And this adjusted true positive rate gives us a good indication of how good are we. This is real life. We've got real samples. We don't know what's in them. And so this is how good we are. And in some samples, it's not very good. We're looking at 12 to 13% true positive rate. I'll talk about what happened there in just a second. In some cases, we're actually getting some pretty good results for about 46%. And when we look at other folks that are in ENTACT, for people who are using similar techniques, our numbers are quite similar. After we've been unblinded, we can then go back and dig in our data and say, now that we know it was there, can we actually see it? So we can go back to our chromatograms and tell it what mass to go look for, see if we can find a peak in that chromatogram and say, yep, there it is. Somehow it just got lost in the data processing. So these are the results after being unblinded. And you'll notice that the adjusted true positive rates go up, which is a good thing. So this is an indication of how good we can be. If everything's working perfectly and we knew what was in the samples, these are the things that we could see. And here we range from about 20% up to about 65%. So we've made some significant improvements there. I think if we can make some adjustments to the workflow, and we've done some of that kind of thing, as well as some of the methods, we can actually make some improvements. So one of the things that happened along the way, in the red boxes, these samples were run late in the sequence of them all, and the instrument had gotten a little bit dirty. The mass accuracy wasn't so great, and so those results were particularly low. We tried a different method, and we were comparing acetonitrile mobile phase to methanol. Methanol was the original method, acetonitrile is the new method, and the acetonitrile results are in parentheses here. And you can see that when we've got a clean instrument, a slightly different method, the results actually bump up a little bit. Now, the interesting thing is the green one was sort of a control case. We didn't do badly on that in the first case, and we didn't do any worse in the second case. There were certainly some discrepancies between the two methods, and we're still trying to sort all that out, but at least we didn't get worse, so that was good news. This slide goes over the reproducibility of results. If you remember back to my evil genius Chris Grolke slide, he did intentionally put things into multiple mixtures to see how well we would do. The different colors are the unadjusted and adjusted true positive rate, or going back to that same idea. So the blue ones are after adjusting for isomers. From a global standpoint, when you don't adjust for isomers, we get 62% reproducibility for the compounds that were spiked multiple times, and the global adjusted average is 75%. It's not perfect, but again, for a first pass, it ain't bad. But again, part of the idea behind ENTACT was just to see where we are. How good can we do? Where are we? How good are these methods? And so we're starting to get into that kind of information now. I realize I'm starting to run out of time and I want to leave a little bit of time for questions, so I'm going to put this slide up and say there's a lot of stuff that we can do with this data once we get it all in. Not only understanding the methods, but pushing them forward, figuring out where the holes are, modeling. Why can't we see this chemical? What classes of chemicals can't we see? Where did these things that we didn't add come from? And how do we know? These are all the types of questions that we can start to answer when we get all the data back from 28 different groups. It's a whole heck of a lot of data, and I feel like if I wanted to, I could probably spend the rest of my career staring at this data. So a long list of things that we can use this data for. Wrapping up, I wanted to let you all know that we are having another workshop, and this one is going to be focused on the ENTACT results. It is going to be happening here in RTP August 13th through 15th. It is free to attend. If you would like to present some research in this area, we're taking basically non-targeted analysis and exposure posters. We'll accept those. If you would like to present something like that, the deadline is July 13th. If you would like to attend, registration is currently open. You can see the website there. It's also been out in a number of different EPA email blasts, so, and feel free to email any of the folks that are listed at the bottom if you can't write it down fast enough. Registration will close for in-person attendees at the end of July and for virtual attendees the week before the workshop. With that, I will put up the smiling faces of my partners in crime, the names of all of the folks that are working on non-targeted analysis research in NERL, not just RTP. Some of the folks are in Cincinnati and Las Vegas as well. With that, I'd like to thank you for your attention.
Great, thank you, Elin. So we'll take first any questions in the room here in RTP.
Have you started to, or have you looked at all at the QC levels of those compounds that no one has observed in any method?
No. Again, we've just barely scratched the surface of this. I did find out something interesting. One of the 195 compounds that we could not see, I was looking through the spreadsheet of what was in each mixture, and it has a concentration that they were aiming for. All of the stuff that's in ToxCap is supposed to be at 20 millimolar to start with, and I found one that it was at 0. And oddly enough, we didn't detect it. And I thought, Chris Grolke really is an evil genius, but in actuality, it's a rounding issue. So the concentration for that particular compound is 0.004 millimolar. But again, we didn't see it, and it's probably a sensitivity issue because the concentration was so much lower than everything else. We just couldn't detect it. So little issues like that are definitely going to crop up. I'm not sure which mixtures those 195 compounds were in, whether they were in the more complicated ones, 9 and 10 at the end, or whether they were in the ones that we should have gotten.
Right.
So that's going to definitely be an interesting perspective to take.
Is there a QA list that's now ready for consumption? Yeah. Very nonchalant with that, Andrew. I've been waiting on that for some time. No, it's been available. Yeah, it's been available. Okay. You can send it around.
Okay, anyone on the line, feel free to unmute your line or write in the chat box. We will take this first question from Joe Binkley. He says, of the 195 not found, how likely are those compounds to have degraded or reacted in vials?
Your guess is as good as mine. I don't know off the top of my head what those 195 chemicals were. That's a list far too long for me to memorize. But as we start to dig into this data a lot deeper, those are great questions that we can ask and start to look into. Thankfully, I'm not the only chemist that's working on this, and we've got some really smart people that are far smarter than me that can look at lists of things and start to come up with reasons why we might not have been able to see those compounds.
So he has a follow-up question. Can you disclose that list?
I think the manuscript that is currently in preparation has that data associated with it. It's certainly not a secret. And so I think when the publication goes out, some of the supplemental material and the background data for producing some of the figures that you saw today will be included, and I'm pretty sure you should be able to tease that information out.
Okay, great. So we'll go ahead and wrap it up for today. Thank you, Elin, for presenting, and for all the participants for joining. Please join us next month for a presentation from Monica Linenbrink. She'll be presenting on EPA's ToxCast Owner's Manual. Have a nice day, everyone. Bye-bye.
The leader has disconnected. The conference will be terminated in 5 minutes.